Hello, everyone. I'm Rob Wright. I'm the chief editor of Life Science Leader magazine. And if you look on the very first slide, we have an image of what the publication looks like. And the reason I put that up there is you actually also get to see what today's keynote speaker looks like, because that's Andy Skibo on the cover of the May 2014 issue. Andy's the Chief Manufacturing Officer at FluGen, an advisor to a number of boards, and consults to AstraZeneca's manufacturing and technology. So say good morning.
Good morning. Hey, Rob, nice to be together on this again. It's a new experiment for me. And as I said when I called, I really hope that you would do this with me to make this go smoothly. So I appreciate it.
Well, you know, it's a circular moment, Andy, because you and I first met at a Bioman Summit back in 2014.
I remember that, and that what turned out to be was supposed to be a 1 hour, let's just have a short debrief interview, I think was most of an afternoon long discussion on state of the industry and capacity planning, et cetera.
Yeah, and so what I wanted to let the Bioman audience out there gain some appreciation for is, look, I'm not a manufacturing expert, and I am not an engineer. But what I really enjoyed about talking with Andy is he had this ability, this knack for communicating real complex problems in a way that it was easily understood. For example, I remember when Andy was describing how a big, enormous, big, large manufacturing facility's got to operate. And he said, it needs to be like deep breathing. Not like everybody's running around with their hair on fire, like you just kicked over an anthill. And I could just picture that. And we're hoping we can deliver some similar insight and visually to you today. So we have a few slides that we're going to show just to kind of help keep us on track on what we wanted to talk about. One housekeeping item, there is a live chat feature. We are going to be paying attention to that. So if you want to ask Andy about like his sailing trip odyssey up the Atlantic, that'd be a good thing to talk about. It's really pretty interesting. If you have more complex questions, we're going to try and answer them. But just in case, we were hoping that for a real long, complex answer, we would put something together in writing and share it with the meeting planners so they could host somewhere on their website. That's what Andy and I kind of thought would be helpful to you. And another housekeeping item we wanted to mention was prior to signing up for this, there was no COVID-19 situation that took place. And so originally we had talked about wanting to cover CAR-T and the supply chain and personalized medicine and the CMO's role in these things. But we thought if we didn't talk about COVID-19, because manufacturing and the supply chain will play a pivotal role in the success of any vaccine that's developed. So we thought we'd talk about that first, and then we'd get into the meat of talking about the supply chain. And then finally, we'll talk about CAR-T and personalized medicine with regard to the biomanufacturing aspect. So with that, Andy, let's spend about 10 minutes on COVID-19 and biomanufacturing, and just maybe you could tell us a little bit about what you're ruminating on with regard to biopharma, and if you could pull up slide 2 right now, you know, kind of what you were worried about maybe 30 days ago versus today versus 30 days from now. So, and let's start with that. Like the first time you and I spoke, we had a lengthy conversation about COVID-19 and biomanufacturing.
Thanks, Rob. And I'll use the same thinking process that I've used for my previous Bioman summits. I like to talk about long range for the industry. In our business, especially in bio, 2 years is tomorrow. That's your locked and loaded manufacturing schedule. So for a lot of our small molecule colleagues, planning ends at 2 years. For bio, we're actually, that's a hard lock on production. To me, 5 to 7 years is coming soon, and 10 years is a planning horizon. You've heard me say that a lot for capacity planning. I also tend to look, my staffs, I think it's a compliment, but sometimes I have to ask, I tend to look at the risks of what could go wrong we're all very good at planning out the details of how things go right. That's why we're in the positions we're in, how all these 27 dimensions of chess connected together and we make a complex delivery schedule. But to me, understanding what are the downsides of if something goes wrong is also an inherent part of planning. And that's not to say you walk away from risk. Lord knows I've been part of big risk steps in my life, including doing my own business. But you should be informed of, if this goes south, will the decisions that I'm making today, 2 or 3 years ahead of it going south, will they look rational? Will we look at that and say, yeah, I understand how you made that decision. That's what we would have done with that data at hand. And at least then we're planning for how to avoid that risk. So for COVID, and all 3 topics today, I'll follow that stream of thought. For COVID, I won't repeat all the things about the virus. I worked in vaccines for 11 years. I've worked in bio since the early 80s. I'm awash in the data on influenza and pandemic. First became aware of COVID, like most people on the science side, in the December, January timeframe. No, it didn't catch us by surprise in March. Its impact perhaps caught us by surprise, but the fact that it was out there and that it was ugly was known for a while. I look more at what happens as we go forward from here. The slide that we put up was just to give an idea of scale and how much this landscape changes, and then I'll talk about my more downstream worries. When Rob, you and I started talking, the slide on the left, 70 some odd players, that was current. That's how many there were just 6-8 weeks ago. Right now, the number is north of 200. Now, some of these players are 5 people in a university laboratory. Others of them are J&J, AstraZeneca, Pfizer, Merck, you name it, with $1 billion and hundreds of people trying to drag this desperately needed product across the finish line. And we looked at things a month or 2 ago. Some of my heads up was, it wasn't the science. In some cases, that's the fast part. It wasn't the clinical trials, and Lord knows that's going to be challenging. It could be dumb things like, if 5 different firms are all looking for a half a billion vials, and they all want it in December, you're just not going to pick up the phone and call the 2 or 3 major manufacturers in the Western world and each order several hundred million vials or syringes. And that type of logjam is going to transpire across the entire supply chain. Doesn't matter whether it's media, it doesn't matter whether it's critical resins for chromatography steps. Every single bit of this will be very different from our normal planning, in that the volumes required are enormous, they're short, and there's multiple players looking for them. It teed up the thought, and this had come up in discussions over the decades with BARDA, of should not everyone can get a half a billion vials. Not everyone's product is going to cross the finish line. Suppose company A is the one that crosses the finish line. Company B doesn't, but they're the one that has the vials. How does the industry decide to trade raw material? Now, I have faith. Bio has traded capacity over the years, small molecule, somewhat less so. Fill finish, quite commonly we trade capacity. I have faith that if raw materials are in plentiful form in company X, but company X doesn't need them immediately and company Y does, I think industry will sort that out. In the interest of the 10 minutes for COVID, let me focus on my 2 big worries. And this is what happens if it all doesn't go right. There's high risk in these hypertrack timeframes. There's high risk in the science. Virtually all of the early products, the earliest ones, the biggest programs, are mRNA, mDNA platforms. That's fast science. That's why they're in the earliest program. It's once you have the sequence, and I know my science colleagues will throw bricks at me, but once you have a sequence, it's relatively straightforward to come up with, if I want this clip of spike protein, how do I get it onto my platform vector? That's why that science advances quickly. But those sciences have a couple of unknowns. One is, while that has been around for a while in multiple places, to my knowledge, there has never been a commercial vaccine launched off that platform. Shows success in monkey models, shows success in other models, but to date, there hasn't been a vaccine launched off that platform. So we're heading into new territory. Functionally, one of the worries, a bit more especially with those vaccines, but for any vaccine, is how do we determine, do we have functional response, not clinical response? To be really simple-minded, if I stick something under your skin, you're going to get an immune system response. What we're really looking for here is, if we produce one of these vaccines, we will get a clinical response, but will that translate into a functional response against the actual virus in the marketplace? That's easier to figure out if you have 2 or 3 years to play around with clinical trials. Merck's, I think, set the record with Ebola, 5 years. And now we're talking 6 months, 9 months, just to put it in perspective. Another issue, again, with mRNA, mDNA, but others, is the durability of the response. And you've already seen one major firm announce, after some of the initial profile, well, gee, we're hoping that our vaccine might last as long as a year. That's kind of the first hand in the air about, let's see how the early data plays out on durability of response. Another issue that is harder to sort out with very short clinical trials is immune system overreaction. There's a bunch of initials for this: ADE, ARDS, there's an even longer one that I can never remember. But in essence, what's happening is the immune system has a memory for the vaccine itself. But if that bit of the spike protein isn't exactly right, perhaps the immune system won't respond to the actual virus when a naive patient is finally exposed to it. The immune system responds to what it remembers of the vaccine. That doesn't work. It notices that it's not holding the virus back, so it responds even more strongly. And I am greatly simplifying immune system response science here. But in essence, what happens is the immune system goes into overdrive. You don't have a final answer on that until you actually have vaccinated people subsequently exposed to the real virus. The last one, which again is harder to determine for these particular platforms, is if you don't have a durability of response, can you give a repeat dose? And in past, for some vaccines, you couldn't do that. If your response didn't last more than 9 months, giving a second dose or a booster didn't work. So these are all things that with 2 or 3 years, we'd sort out. We're not going to have the chance to sort all that out ahead of time. And we're going to be, the possibility will be in tens, hundreds of millions of patients before some of these answers get answered. So what is the risk? Why do I worry about that? 2 things. One, in the past, where industry's gotten itself in trouble sometimes, usually a single industry, a single player, impeccable credentials, decades of perfect experience, and then they get in trouble. If you look at a lot of those instances, what's happened is so much of the corporate profile was invested in the success of that particular product. It was going to save the P&L. It was going to drop another $2 to EPS. It was going to be breakthrough therapy, whatever. So much of the profile got invested in the success of that product that when they began to see signals that the product may have had an issue, it was very hard for the corporation to admit it. And you'd spend a year or two massaging, now we're going to have years on this one, but you're going to spend a year or two massaging the data. Gee, that's bad news. I've got my profile out there. Can I really turn around and say my shot on goal didn't work? That's one. The other one is I worry about the vaccine industry and the impact with all the first hype. And there's not a lot of shading. I'll be really frank here. There's not a lot of shading on the hype of the fact some of this may not work. Not all of these products are going to cross the finish line. Right now, we have Congress, we have other national governments all hearing, I'm going to deliver 500 million units in December, period, end of discussion. Not might, or not maybe, or not subject to, but I will. Suppose we don't. We could turn success into negative. We could get 10, 20, 30 million initially out there, but it's not 500 million. Or we could get 4 shots on goal, 2 of them work, 2 of them don't, but with all this press, that will perhaps be perceived as a failure. And as you and I talk, Rob, I'm really disappointed with the populist streak. It's not only the US, it's the UK, it's Europe, this populist streak, this anti-vaccine, anti-science streak. If we wind up with a more difficult start of developing the vaccine, or worse yet, one goes out and then we find out after lots of people have tried it, that there is a side effect, are we as an industry going to throw gasoline on that fire? So I'd like a little more temperance in the message around this. So that's my spiel on COVID.
All right. You know, one thing, and we did spend a little bit more time on COVID than we wanted, but there was one thing I did want to ask you about, and that was, because I saw we talked about where you're going to get the glass, but I saw a press release about the government signing a deal with the manufacturer of a glass-like material. And I remember you had an experience where, an issue in manufacturing where something was changed in the drug and they couldn't track what happened or where it was. Can you share that real quick?
And it is, and I'll try and be brief, but to me, it's one of the highlights of, in bio, we're developing new science all the time. So one half of this is pure change, full speed ahead, invent new business. We're all good at that. We wouldn't be where we are without it. But once you have it in manufacturing, the other half of your brain kicks in, because change is bad, especially change you don't know about. I always joke, if the plant manager of my big site parks 3 doors to the left of the main entrance, I want him to always park 3 spots to the left of the main. I don't ever want him to even move his parking spot, because change has odd ways of creeping into the system. The one you're talking about is a great example. It goes back while I was still at Amgen, and I remember it was March of a year. I don't remember the year, but March will stick in my head for a long time, because we began to lose 20 to 30% of 2 key products due to protein aggregation in the 3 ml syringe that it was in. And you do the usual things. First, you go, is this drug substance? Is there a contaminant, fibers, everything? We tore the process upside down. We looked at the science behind it. We looked fill finish. We went back to, could not find the issue. You talk to your suppliers. Have you changed anything? Everyone swears on their grandmother's grave they haven't. They're required to notify you if they do. 9 months of losing that kind of volume, and fortunately we had enough resilience in the system that didn't resolve a market short, although it was tough. Finally, we picked up the phone and called our colleagues at Genentech and said, guys, have you had protein aggregation in syringe only product? Yeah, we have. And have you found the answer? Your products are different than ours, processes overlap. No. When did it start? March. And the light bulbs go on. It has to be a supplier issue, because there's too much that are different in the 2 plants on different products. So we combed the list, went to every major supplier, and we landed on the vendor of the syringe, and I'm not going to name the name, but it's easy to guess which 1 or 2 or 3 it might be. Had you changed anything? No, absolutely not. Same plant, same people, same raw material, blah, blah. Something changed, and the key was it changed in March. Whatever you were doing to the syringes that we both got in March, that's what changed. And what it was the hole in the end of the syringe is made by sticking a needle into the molten glass. And that needle was originally made out of titanium. And a good manufacturing thought was tantalum's even tougher. So let's get rid of the titanium needle and we'll have a tantalum needle. That was the change made. One of us had worked on float glass for the automotive industry when they changed from rolled plate glass to glass made flat by floating it out on tin. And for months, there was this unexplained 50% of the product really was difficult, had quality issues. And we determined that the part that touched the tin didn't bind the same way to the plastic. Bingo. There were a few atoms of tantalum is in the same group as tin. There were a few atoms of tantalum sticking electrostatically to the pores of the molten glass. You pick up the phone and call our colleagues in R&D and say, if we had a few atoms of tantalum in our glass, what would happen? Oh, that's easy. It's going to cause protein aggregation. Bingo. Please go back to the titanium. Who would have ever have put that together? So that's why I say when I see something like this is we'll just substitute the glass. Yeah. Are you going to do that in 3 months? Will we know what that does 6 months from now, a year from now? And I just pick that change out of the air. Any of the others that are offering solutions to this crisis have to be evaluated with that kind of mindset. You just don't make a swap in August because it's the only key to the lock.
Right, right. So Andy, since we were talking about supply chain, let's transition, over to the supply chain issues that we wanted to talk about. Like, what have you been stewing over with regard to that? Like trade policies, and when we talk about planning for this, we were talking about, biopharma as a strategic asset. There's a lot of different things to talk about, but maybe you could provide for this audience some of your thoughts around the importance of the supply chain as we move forward into the next 5 to 10 years.
No, and again, this is another, I'll be frank one. What really teed this up, and it happened within the last month, as you know, Rob, we weren't talking about this 2 months ago, was we're beginning now to see the consultancies stepping in to help all of industry, but especially pharma, because I get those calls, on how do we adjust our supply chain for the changes that have occurred thanks to COVID, thanks to trade policy. In other words, it's becoming more difficult to bet significant pieces of your supply chain, whether it's raw materials or actual product, on China, India especially, but others. And what really made my face turn a little red was this was being talked about as though this was a new problem that had just come over the transom and now we'll figure out how to fix it. For the audience that's on this call that was part of Bioman earlier summits, at least 4 or 5 years ago, I teed up, Lance Miner helped me do it, a discussion on the risks that we were taking in pharmaceutical supply. And we talked about how we had leaned out our supply chains. Everyone, if I hear the Toyota model one more time, I'm going to choke. But we had all leaned out our supply chains. In the 80s, when I started in this business, I'll pull up a couple of those old slides for the next time I do one of these, but we'll need more than 10 minutes. 50-some-odd% utilization was typical pharma network, 50%. Today, the ideal is 85 to 90. All that fat has to be gone. It was all internal. Any major product usually had 2 active, separate supply chains, even for the raw materials. The raw materials were internal. It wasn't uncommon to have 3 separate supply chains and 2 older ones, so that if one burned to the ground, you had as many as 4 or 5 backup options that you could. Now that cost money. I can argue with cost of goods manufacturing of 12%, 17% for bio, 7%, 10%. Just how cheap does the product have to be? Another story. It was necessary to change those supply chains because the industry probably wouldn't have survived in its current form if we hadn't. There was a great slide by McKinsey that normalized earnings earnings total just kept it at 100. Earnings grew by about almost double in the 12 years of the study. R&D costs, earnings actually as a percent of total went up. So did R&D cost. So did S&OP cost. So did clinical trial costs by a lot. So, quite frankly, did depreciation. So the thought that we weren't building plants in that 12 years is wrong. Depreciation. So how do all your base costs go up, but earnings as a percent of the total actually grows? And the answer was manufacturing supply chain stripped out almost 50% of the cost of manufacturing in those years. So the guys that were hired to run supply chain from apparel, from automotive, from footwear, where pennies count, they earned their keep. But that was in a period where the industry had no pipeline. You could predict volumes pretty easily. The last 5 years, we've had pipeline. You launch a new product, you don't have 6 months to get the launch wrong if you get an accelerated approval. You better have product. We pride ourselves in bio, and I think small molecule and AZ is the same. The first 3 big bios we launched in the last couple of years, the product was in the hands of a physician within 24 hours of license. That requires resilience. That requires a spare pack line that's just sitting there waiting for you to get the package insert ready to go. That's very difficult to do in a pure CMO network. Very difficult to do if you have only 1 node instead of 3 nodes. In bio, we kept 1 plant at 60% utilization just to allow for that. So the reason for this long-winded spiel is I'll ask both us, our industry, and our big consultancies colleagues to remember to include resilience and to remember to include downside of risk in the equation. Those factors are in most mathematical models of supply chain. For 2 decades, we've set them at 1, which basically takes them out of the equation. Not delivering a new model of a Toyota for 6 months is not the same as not delivering Infinzi for 6 months.
So Andy, let me ask you a question because I'm just keeping a track on time.
Yeah.
One of the things we talked about with regard to the supply chain, we want to be real quick about. Slide 6 brings up plasmids. And we talked about one of the challenges of regulation of a raw material like plasmids being regulated like a raw material. And under slide 5, it kind of shows the, we call this the firecracker slide with the warhead. Could you briefly explain, in about 60 seconds, a bit of the aspects around the torpedo warhead and the regulation of such?
Yeah, this got my attention in a couple of consulting assignments as I began to move into this industry and be asked to contribute to it. And I don't want to lecture on the science here. There's people that understand it a lot better than I do. But I think what I wanted to make sure we got off the firecracker slide was the fact that the product actually has a vector, and I call it a warhead. The effect part is what it's labeled on one of these slides, and the guiding part to me is the torpedo. Plasmids are a piece of that, and we have a slide that shows what a plasmid looks like, but plasmids are a piece of that essentially is used in the product and goes untransposed directly into the patient's cells. So as I looked at this CAR T, 2 things struck me. We'll have the firecracker slide, but if we can, we had the manufacturing CAR T cells, which showed the whole loop of supply chain. So the 2 things that I wanted to take away from our discussion today are the supply chain to me looks like half pharma and half Amazon. Let me finish that thought and then I'll come back to this plasmid because that's a regulatory affairs piece. If I were hiring for a new plant, for new manufacturing, I'd hire one of me when I was 30, but I'd also hire one of the best supply chain people out of Amazon or similar that I could hire. The product comes from Jane Doe. Let's call that vial number 120, patient 123. Patient 123's vial in cryo has to move through cold chain supply to our plant, 24 hours, 48 hours.
Slide 7, show that, kind of show that if you pull up slide 7. Keep going, Andy.
Okay. That to me, the blood fractionation folks do that part so far today. They do track individual patient bag of plasma up to the pool stage. And that's where it stops. After the pool stage, it's like the rest of pharma. Unlike the rest of pharma, each step down the plant, not only will we have to isolate and quarantine a particular process step, we're going to have to isolate and quarantine patient 123's particular process step. And at the very end, when we have the final product for patient 123, we have to track that supply chain, cryo chain likely, all the way back to patient 123. We have to guarantee that that's there. And what struck me about the Amazon model, with all of us spending more time at home, had enough time to waste to watch a book actually delivered, and watched Amazon tell me warehouse by warehouse, truck by truck. The last one was, it will be on your doorstep between 2:30 and 3. And lo and behold, that's when the book showed up. That's the piece we'll have to add. If we jump over to the plasmids, the fun of inventing a new industry in our piece of the business is you have to invent everything. Science invents the product. Clinical trial folks figure out, how do I run clinical trials to arguably prove it works. Then it moves into our shop and our regulatory affairs shop. We have to make the manufacturing process. That's fun. All this is from scratch. This is like bio in 1982 when we were moving into deep tank and nobody thought you could do it for mammalian. And we have to work with our regulatory colleagues to figure out, how are you going to regulate this? And the plasmid bit was, to me, one of the highlights of, we haven't figured this out yet. Plasmids started as something that, and still to this day, our R&D colleague can buy out of a catalog, or you custom order the plasmids you want. Are they CGMP controlled or not? They don't have to be for R&D. I've seen CGMP light. I've seen what folks are calling full CGMP. I've seen the same company buying all 3 versions of the same plasmid. And I'm wondering what kind of controls are back in the home office to make sure that the R&D version of it doesn't cross the CGMP version. Worse yet, the CGMP light versus the CGMP real. The bigger thing that stuck out to me was they're still regulated as a raw material. They're not regulated any differently than a media component. And yet, if you go back to the firecracker slide, that plasmid goes straight into your cells. I just don't think that in the long haul, when our regulatory colleagues get their heads around this, that's going to remain regulated as a raw material. Is that a co-product? And the reason I tee this up is sometimes for a startup firms, especially pure science driven, the first reaction is to push back on regulatory affairs, push back on the regulators. Oh, they don't know what you're talking about. This is new science. We're way ahead of them. Believe me, I sat in those conversations in the beginning days of bio. That does not make friends with the FDA. That's a very quick way of finding out that regulator is a capital R and regulate T is in 2-point type. So what we want to do is what we did with bio, is you become partners with your regulators. And if you think through, how should this be regulated? Not how is it easy to regulate, but how should this, all of us having the experience we have, if we were properly regulating this and this product was going into our kid or grandkid, how should we regulate it? And the firms that get that right, and I hope Genentech doesn't mind me saying it, but they were one of the breakthrough companies. They were thought to be a little more expensive than what we were building into those plants, but we wanted the regulatory aspects of it to be bulletproof. And we worked with our colleagues in the agencies to jointly develop what that had to be.
So Andy, we're at the end of time, and I'm just curious. It's a unique opportunity for us to present to Bioman. Do you have a parting pearl that you'd want to offer to attendees today?
Yeah, two things. One, you listen to a lot of what I've talked about. I think if we at our, especially those of us in senior positions, but any rank can push this thought, we should always look at, are we managing whatever part of this step we're in the right way, including the positioning of it. We don't want our industry to be colored with, these are a bunch of cowboys and the chickens came home to roost. I'm making mixed analogies. So that's one. The other part of it is, think of it as a whole. Science, development, commercial, clean ops, manufacturing, and whether you're in the new part, the CAR T, personalized medicine, think of it as a whole, even if you're only responsible for the manufacturing piece. That will save things like missing resilience or missing the risk equation on supply chain, which is pretty cut and dried stuff. That will help as we have to invent an industry from scratch, like CAR T or reg affairs.
So Andy, thank you so much for making time for us today. Thanks for inviting me to moderate and sharing your wisdom. And thanks to the sponsors of today's summit.
Great. Thanks, Rob.